Hello everyone, welcome to the NN Classroom. Today, I'd like to share with you a multiple choice question number 30, extracted from the Specimen paper 2016, concerning the topic in topic 8, metabolism, especially in 8.1, enzymatic inhibition. Let's see, asking for how does a competitive inhibitor, main point here, competitive inhibitor, so which means not non-competitive, right? While interacting with the enzyme. First of all, people should have realized that, a student should have realized that competitive here, which means the inhibitor is going to be competing with the substrate. That means there's a competition between the inhibitor and the substrate. And as we know, substrate is the one that binds to the active site of the enzyme. So that's why if you have a competition, means that both you, inhibitor, and the substrate are in a competition versus the substrate, right? There is a competition between you, substrate, and me, inhibitor, in chasing to bind the enzyme. So as we know, substrate binds to enzyme in active site, so meaning that no doubt your inhibitor is going to be bind to the enzyme, binds to the same site, right? Otherwise you won't be said to be competitive, right? So that's why both bind to the active site, and as we know, substrate enzyme must be interacting within the active site. So that's why first of all we can sort out C and D cannot be the right choice, because the one bind to the allosteric site is what we call bind by non-competitive inhibitor, right? As there's no competition, so that's why the inhibitor binds to one site and the substrate binds to another site, which they are not interrelated, right? So that's why answer only be sorted out as A and B. Next thing is that whether the inhibitor will able to denature the enzyme, and this is not really, right? That means whether competitive inhibitor can denature or not denaturing the enzyme depends on another factor. The factor which is that whether the inhibitor is a reversible binder or an irreversible binder. So this is another concept. So that means to be just looking at it is a competitive inhibitor, it doesn't tell whether this inhibitor will be so powerful that kill or denature the enzyme or still letting the enzyme to be intact, right? So that's why the word competitive only says you are competing with the substrate, but whether you can or cannot denature the enzyme depends on the reversibility. Here, depends on the reversibility. Yeah, whether it is a reversible binder, then you won't denature the enzyme, or it is irreversible. That means you can denature the enzyme. So that's why for here, if you just say competitive, we do not know. We do not know whether you can or cannot denature the enzyme. So that's why A is not a good choice, right? But B is a right description, because once you compete means you prevent the substrate from binding to the same site again. As you have a certain concentration of inhibitor that will block the binding of the substrate to the enzyme. So that is always meaning the competition, right? So that's why for here, this sentence is an elaboration. Is an elaboration explaining further what is meant by competitive, right? They are both competing in binding to the active site. So that's why answer goes to B, a right description.